the EU regulations to the implementation of the Cartagena Protocol. He is currently a professor of biotechnology regulation at both Ghent University and Free University of Brussels, having advised over 50 governments, including Egypt, by the way, on the complex scientific and legal aspects of biotechnology and environmental policy. He played a significant role in modernizing Europe's biosafety landscape, specifically managing the project to implement national biosafety framework for 10 EU pre-accession. Dr. van der Meer's lecture will give us a global perspective on regulations related to gene-edited organisms.
Thank you very much for your kind introduction. Let me first check, can you hear me?
Yes, sir, we can hear you. Go ahead, please.
Wonderful. Let me start sharing my slides. While that starts up, I trust that you will let me know if the slides are not visible. So here we start. Should be in presentation mode now. First of all, my thanks to the organizers for inviting me to talk about the global discussion on the regulatory status of gene-edited organisms, and also my thanks to the to the previous speakers for some excellent presentations on the science and the practical applications of gene editing. Makes my life much easier. So what I intend to do in the next 25 minutes or so, give you a little bit of an idea of the context. Why do we have the global discussion on the regulatory status of gene-edited organisms, and where is that discussion now? The context is this. It is what I refer to in my law classes as the regulatory cycle. And every regulator knows exactly what that means in practice. It means that they start developing a regulatory framework. And for many of us, it was 40, 50 years ago. And after that has been developed, and there is typically a period of 5 to 10 years of implementation, compliance, and that contains many elements, risk assessment, decision-making, enforcement, public information, and so forth. And then typically, regulators, governments review and assess the systems after a couple of years. And that may be 5 years, that may be 10 years. Sometimes this is just done as an initiative from the government. Very often there is an obligation in the laws themselves that require governments to review the system. And the discussion about the regulatory status of gene-edited organisms is part of this last part, the review and assessment of regulatory framework. And to complement this whole picture, if the review and assessment indicate that there is something not working with the implementation, then adjustments are made. If the review and assessment shows that there is something not clear in the regulatory frameworks, then that is used to make adjustments. And this is a continuous process. This is why we call the regulatory cycle. Regulators do not do this once every 5 years, they do it just every day. Whenever they see something coming up that doesn't work well, then they will make a note of it and they'll try to improve it. So as I said, the discussion on the gene-edited organisms and their regulatory status is started as part of this review and the assessment. Let me briefly summarize. So review and assessment are different things, and you see this in some international agreements, articles about review and assessment. Typically, we refer to review of the legal text as it is written, whereas assessment is more the assessment of the way it's implemented. And I'm going to focus on the review of the legal text. And reviewing the legal text of any regulatory system follows a number of standard regulatory, let's say, quality parameters. And the first one, of course, is it still clear? Is it the justification? Why do we have these rules? And in some jurisdictions, we refer to that as the intervention logic. Why does this particular field need to be subject to regulations? And if you're going to change the rules, why are those rules going to be revised, or why do we need rules? So that is all as part of the first parameter of the regulatory review. Then a very important one is clarity. And that is typically the question whether the objective and the scope of the regulations are still clear, and especially if they're still clear in light of new technological developments. And to give you an example, whoever wrote about 100 years ago the traffic regulations could not have foreseen that one day there would be a Google self-driving car. because the rules then made very clear, if you drive a car, then you are responsible. You're responsible if you're speeding, then you get a ticket. The new developments, there are cars that drive by themselves. And then the question comes up, who gets a speeding ticket? Is it the owner of the car? Is it the owner of the software? Is that Google? Is it the owner of the camera? Whatever it is. So this means that new technological developments always constantly force regulators to think how the regulations relate to those new technological developments or the other way around, and whether specific rules are needed or adjustments should be made. Other parameters are consistency. If you have developed your regulatory system 10, 20 years ago, and in the meantime, your country has joined some international agreements, then it needs to be checked whether those regulations are still consistent with the international agreements. Are they still consistent with other national rules? And of course, are they still internally consistent? Question is also, is a system transparent enough? This means, is the decision-making process predictable enough? Is it proportional? Is it workable? Other regulatory parameters are, is it enforceable? Regulations that are not enforceable are not very helpful, because it leads to a loss of credibility of the system. And the last question that is always important to look at, since biotechnology changes so often and so frequently in directions, is the system adaptable enough? So these are the type of questions that are being raised when you do a review of the text according to the regulatory quality practice. Now, the discussion about gene-edited organism came up under this point, and it was already a couple of decades ago when that started. But there's the question whether the objective and the scope was still clear enough in relation to a new technological development, and the new technological development then was referred to new breeding techniques or new genomic techniques. And why was that a discussion? It started around 2005, and there was a discussion about these new techniques, such as gene editing. Why? Because those techniques can result in genetic alterations that also happen in nature, or that can also be obtained with conventional breeding. And then the question arose, and we didn't have that question before, because before, when you would use agrobacterium to do a transformation, then by definition you made something new, because you inserted a whole cassette, for example. But if you can use gene editing to make some small changes that happen in nature, then the question arises, and that did arise, do those organisms fall under the regulations, and to what extent? Do all of them fall or only a subcategory? And then I'm talking about the regulations and the regulatory definitions of GEO, engineered organisms, as they refer to in the United States, or GMOs, as they refer to in Europe and a number of other countries, or LMOs, living modified organisms, as are referred to in the Cartagena Protocol on Biosafety, and many countries that have used those definitions of the protocol. Quick step back, new breeding techniques, new genomic techniques, it's an umbrella term. It's a large variety of techniques are being discussed under that. And unfortunately, not all of them are actually new, or not all of them are actually breeding. But the common factor is that these are techniques that can result in something that is unlikely to happen in nature with conventional breeding, and it can result in something that does happen in nature or with conventional breeding. One of those techniques is RNA-directed DNA methylation. There is a technique, grafting on GM rootstock. You're all familiar with this. To what extent are non-GM parts GM rootstock? Reverse breeding is a very complex tool, and breeding very useful tool. Expression. To what extent are plants that have been, for example, treated to express a certain protein, for example, but in a transient way, for example, by drops on the leaves, to what extent do those result in GMOs? The cisgenetics, and then gene editing or genome editing. And I'm focusing here because this is what the discussion of today is about, gene editing. And then again, in gene editing, that is not one thing, but it's a whole collection of tools. The key thing is the word targeted. Where in the past, the transformations with agrobacterium were very specific, because instead of mixing thousands of genes, you introduced just a few genes, but it was not very targeted, because scientists could not predict where the gene would end up in the chromosome and on which chromosome. With gene editing, that has become a very different thing. it's very targeted. So it's not only specific, but it's very, very targeted. There are several different types of genome editing. And then we have, of course, the ODM type of genome editing. Then we have site-directed nuclease types of genome editing. And I'm just listing a few here: zinc finger, TALEN, and as has been discussed several times in this seminar, CRISPR-Cas. So I'm focusing on CRISPR-Cas now. Fortunately, with the excellent presentations before me, I don't have to discuss much detail, but the summary of it is that there is a mechanism that makes a double-stranded break, and then there are different ways in repairing that break. Either it can just be done in a random way by non-homologous end joining, or you apply a template, and then you have homologous end joining, homology-directed repair. Just one or two base pairs, and it can be an entire gene. So this is the situation regulators were confronted with when we're having these discussions. And what can be the result of these? It really happens, as the previous slide showed, what happens after the DNA cleavage, after the double-stranded break. You can make very short random deletions or insertions, indels, and they can result in a loss of function of a target gene. We refer to it as a knockout, and typically this is referred to as SDN1. So it is a targeted change, but the actual change is a random one because it is a double-stranded break is made, or sometimes a single-stranded break, and it is in a random way. A few base pairs are added to it or eaten away. This is what we call SDN1. Then you can make very small changes, but in a directed way by using a template. And this you use the homologous DNA repair, as has been discussed before. And there you could refer to as correcting a genetic disease. Let's say that a few base pairs are missing, so you knock it in as opposed to knocking it out. And then the third category is this SDN3, whereby you use the homologous repair mechanism to introduce entire genes. This you can do too. It all depends on the length and the details of the template you're using. So instead of thinking about gene editing as just one thing covers everything, no, gene editing can result in very different things. Very small knockouts, very small knock-ins with a template, and you can introduce entirely new genes. You can also introduce genes that are already available in the gene pool by using SDN3, but just in a much more targeted and precise way. And confronted with this picture, and remember, this is only one technique of gene editing, and gene editing is only one of the many techniques that are discussed on the new genomic techniques. But this was the main focus of the debate, simply because this is being used at a very large numbers at the moment worldwide, as the earlier presentations have already shown. So confronted with these cases, the question arose for regulators. Now, do these resulting plants, we are talking about plants mostly, do they fall under the existing definitions? And if so, to what extent? And that debate has been in many jurisdictions, it has been finalized. And it actually depends on what the result is. And the result on the regulatory level is a differentiated picture. In most cases, SDN1 and SDN2 are not covered by the regulatory definitions. This is not in all cases, but this is most of the SDN1 and SDN2 are covered. Whereas most SDN3 are covered by the regulatory definitions. So as we see in the previous slide, let me just go back. Depending on what is the outcome of the whole process, SDN1, 2, or 3, in many jurisdictions where these discussions have been worked out, and sometimes they lasted 10, 15 years, they come to a sort of a differentiated picture. And that differentiation is that single applications of SDN1 and sometimes also SDN2 are not covered by the regulatory definitions, whereas in most cases, SDN3 applications are covered. Then, of course, is the next question is how do you reflect this in regulatory terms? There are different options to do that. The first one is just you issue a clarification. You just don't change anything, but as, for example, the Philippines did, they explained that looking at their own definitions, there's an explanatory document that explains that in most cases, SDN1 and SDN2 are not covered by the regulatory definitions, whereas these are. Then there's a second approach, a very interesting approach. It's a clarification. So the law and the definitions were not changed, but there's an additional rule. And that rule says that In these cases, these organisms are not covered by the definition, SDN1 and SDN2, but it is not you as the applicant yourself who decides that finally. You have to submit your conclusion to the authorities, and they will look at the question whether you have correctly concluded where these organisms, let's say an SDN1 application, does not fall under the definition. This is, for example, the case in Brazil and Argentina and a few other countries. So the law, the definitions have stayed the same, but they added an additional rule that allows the governments to confirm your assumption if a certain organism, as the N1 organism, for example, is not covered by the regulatory definitions. Then there are also cases where the rules had to be changed. So the conclusion was that these are not covered or should not be covered, but it may very well be that the rules are not clear enough. Now, there are two ways to change the rules. Either you keep the definitions as they are, for example, and they would lead to the conclusion that an SDN1 application is covered, but you can exempt those categories. So rather than changing the definitions, you can just leave them as they are, and then clarify in an exemption that certain categories, let's say SDN1 or certain SDN1, covered by the regulations. This was, for example, done in a new rule, what they call the secure rule of the United States, USDA APHIS, where they kept definitions as they were, but they clarified that exempted were certain SDN1 applications. That regulation in the meantime has been nullified, not for this reason, but for other reasons by a court. So they're now working on the old system again. But it's a good example how you can keep the definitions as they are. And in order to achieve this differentiation, you can just exempt. And then, of course, the last one is you just adjust the definitions. If you come to conclusions that you believe as a government that as certain SDN1 and SDN2 applications should not be covered by the regulations and your definitions are ambivalent about that, then you just sharpen the definitions. And one way to adjust the definitions, I'm quite pleased to see that, is that a number of countries have adopted the definition of the Cartagena Protocol on biosafety, because that definition makes very clear in which cases these organisms are covered and in which cases these organisms are not covered. I have left it with this general overview as to what are the general conclusions, i.e. differentiation, and what are the different ways to, as a government, to clarify that differentiations. I will not go into detailed examples because I know speakers after me will do that. I thought I'd keep this brief, and I'm happy to answer any questions because I think this is it. These are my email addresses from the universities where I teach, so feel free to send me any further requests for clarifications or examples, and I'll be happy to do that. And I hope I've kept this short enough to allow for some questions, if any. And I will stop sharing here so that I can see you again. And I hand back to the chairman. I have no sound.
Thanks, Dr. Bendermeer. I think we will hold the questions until the end of the session. So
Okay.
Thanks again for your excellent presentation. So our next speaker is Mr. John Coleman.
Yes, and I hope you can hear me.
Yes, I do.
Excellent. Let me start sharing my screen. Let me know if it's working properly.
Yes, we can see. Yes, we can see you.
Perfect. Well, good afternoon. Yes, go ahead, please. Sorry.
If you want to go ahead, that's fine. So just give me a minute to introduce you properly.
Yes, please.
Okay, so I'm happy to introduce our next speaker, Mr. John Coleman, an international policy advisor and the Africa coordinator for the Program for Biosafety Systems. With over 30 years of experience, John specializes in biosafety regulations, IP management, and international technology transfer. Having advised organizations ranging from the Stockholm Environment Institute to the World Bank, he brings an unparalleled depth of experience in helping partner countries develop workable and transparent regulatory systems for agricultural biotechnology. His strategic guidance has been instrumental in the success of major initiatives for the FAO, UNEP, and the Gates Foundation. Mr. Coleman, the floor is yours.
Thank you once again. And let me share my PowerPoint slides. I think it's up. Yeah. And well, in addition to the work that I've been doing for the past 20 plus years with the program for biosafety systems, more recently, I've been working with my colleague Benno van der Laan, who's based in Maryland, in close to Washington, D.C., who's running a small consultancy firm called Green Orange. And Benno has been implementing for the last 10 years or so the USEC Emerging Markets Program, with USEC being the US Soybean Export Council. And through the USEC Emerging Markets Program, as well as the Program for Biosafety Systems, I've been interacting with a range of countries and governments on the African continent, including Egypt, and on and off to discuss regulation of modern agricultural technologies, including gene modification and more recently genome editing, of course. And some of the findings and results of that work will be used in this presentation. So thanks once again to Professor Nagla for inviting me and Benno to be involved in this important workshop, and also many thanks to our partners such as Agiri, ICARDA, and all the other local, regional, and international organizations represented in this meeting. It's a pleasure to be here. And we're slowly moving from the global picture to the regional picture, and sharing experiences as to how African countries have accessed this new toolbox of genome editing, and how they have approached the regulatory aspects of genome editing. So yes, I'm also benefiting from being one of the last speakers in a row, because many of the points I'm making will also have been illustrated by previous speakers, including Professor Nagla, Lina Tripathi, of course, as well as Professor Piet van der Meer. So we all know by now, early on in this workshop, that this set of technologies is rapidly moving from hype to reality, with a range of products that are actually commercially available in different markets. Non-browning bananas were already mentioned. Gene-edited mushrooms, the GABA tomato, which is a healthier tomato, which has been approved by Japan already a few years ago. Puffer fish was mentioned, also approved in Japan. And what's important with these products is that they are not regulated as genetically modified organisms. That is not to say actually that they're unregulated, but they're regulated not as GMOs, but as more as conventional varieties and conventional products. So even as I was preparing for this presentation in Egypt, even in this month, lots of new news items came up that illustrate the points that have been made already in this workshop, and that will be made in the course that will be following this Sunday workshop. And that is an approval for gene-edited non-browning potato in Japan, which I already mentioned, but also it shows that things are moving in Europe as well, with green light for field trials of gene-edited barley. And this is significant because it falls within the scope of a new act in the UK, the Precision Breeding Act, which is a new legal mechanism which is actually targeting the types of new genomic techniques or precision breeding that has been discussed in many European countries, as well as in the European Union for many years now. But the UK is definitely the first country to go ahead. And I think that's also influencing the discussions in the European Union. As you know, after Brexit, the UK is no longer a member country, but also in the European Union, the picture is definitely changing. Still remains to be seen what the final definite outcome will be. But it looks like, as Lena also mentioned and others have mentioned, that applications of SDN1 will not be regulated as GMOs, whereas other more complex applications will be. This should be a topic of an entirely separate presentation, so I will not be dwelling on that for too long, also for the sake of time. And when I say that these new products are not regulated as GMOs, I think it's important to for all of us to realize that we're talking of an expansion of the plant breeding toolbox, really based on a strong foundation of crossbreeding, mutation breeding, as well as genetic modification. Many countries and research institutes are now embracing gene editing and are thereby expanding the plant breeding toolbox, but it should really be seen as a continuum of technologies and tools and not something as self-standing. And this is also translated in some of the policy approaches and policy decisions that I will be showing later on. Countries that have a strong basis, strong foundation in conventional plant breeding, as well as in modern biotech and genetic modification, are the leaders on the African continent in accessing as well as regulating applications of gene editing. So if we look quickly at that foundation, this is the current status of genetic modification crop development on the African continent. This is a recent overview from AUDA NEPAD, the African Biosafety Network of Expertise, where we see that in East Africa, Kenya, and Ethiopia are advancing relatively quickly on the GM front with commercial releases of genetically modified cotton and maize and cassava. Some of those in Kenya are still held up in court procedures, but we're expecting decisions on that sooner rather than later. In West Africa, Nigeria and Ghana are the leading countries in adopting genetically modified crops. In Nigeria, there's 3 genetically modified crops right now that are planted commercially on farmers' fields, including genetically modified insect-resistant cotton, pot borer-resistant cowpea, very important food crop across the country. And more recently, Tela maize, drought-tolerant maize, has been approved for cultivation purposes as well. And all these crops are performing extremely well in Nigeria. In South Africa, of course, has a track record of more than 20 years in cultivation of genetic modified crops, and has been leading the pack in adopting cotton, maize, as well as soybeans on the continent. And those are the countries that have also been debating applications of genome editing and how the regulatory aspect should be considered. What we do see is that, with the exception of South Africa, the actual rates of adoption are still very, very limited of GM crops in African countries. And there's still a lot of room for expansion in terms of the adoption, commercial adoption of GM crops in African countries. So although it's an evolving picture, the actual situation right now is that adoption of genetically modified crops is still extremely limited across the African continent. So if we turn to gene-edited crops, the main point, as already shown by previous speakers, is that The research pipeline is very rapidly evolving. A recent review published by Science Direct has I think, an excellent overview of what's in the research, as well as field trial stages, and also some recent products where research has been completed and where new products are actually ready for market, such as waxy corn, but also drought and salt tolerant soybean, and so on and so forth. And again, if we compare this to genetically modified plants, the pipeline as well as the actual product portfolio right now is so much more diverse in terms of the types of crops that are involved, as well as the types of traits that that are targeted in this type of research. So these examples were already shown by previous speakers. These are some examples of products that are on the market in different countries and different parts of the world. And again, it's important to emphasize that these crops are regulated by the usual IP and variety registration regulations, but they are not regulated as GMOs in the countries where they have been launched. If we come a little bit closer to home and look at developments targeted at African countries, this is a recent analysis that I contributed to with IFPRI colleagues, with colleagues at the International Food Policy Research Institute, looking at the types of projects that are underway in CGIAR centers. As you know, ICARDA, IITA are among the CGIAR centers. These are international agriculture research centers that work with national research organizations as well as advanced research organizations globally in developing crops that are relevant to tropical agriculture and specifically to African economies. So Lena gave examples from what IITA is working on. Those are important food security crops that are now being improved with the applications of genome editing. And as you can see, again, there's a wide range of crops involved, as well as a wide range of traits that are being targeted by these international centers that have a presence in on the African continent as well. And I think this is important not just for national research organizations to get access to those technologies, but also as a conduit to collaboration with advanced public and private research institutes worldwide that have the intellectual property over these technologies and can make them available free of charge or at limited cost to the emerging economies on the African continent. So if we look at how these technologies and products are regulated, again, these are some findings of a recent analysis that I contributed to, conducted by IFPRI, where we looked at how different African countries are regulating genome editing in their legal frameworks. And it's important to acknowledge that the goal of every regulatory system for new technologies must be future-proof. It must be narrow and specific enough to be clear, and broad enough to cover new technologies as they emerge. And this is very much dependent on the biosafety laws and other relevant laws that are already available in those countries, as well as the overall regulatory philosophy of a specific country, whether it's restrictive in terms of allowing new technologies in agriculture or much more permissive. And it's important to note that a growing number of African countries are proactively developing or have proactively developed their policies on genome editing. So some have genome-specific guidelines developed and published, and we'll go over some examples later on. Some have modified their existing GMO biosafety laws without necessarily developing new guidelines. And some countries are in the development of either defining their genome editing policy or have a set of guidelines underway. South Africa is a good example. I will not be dwelling on that too much, where government felt that the existing GMO law was sufficient, and have adopted actually a rather restrictive approach to genome editing, equating it with GMOs. So all applications of genome editing are so far treated as genetic modification in the South African context. But other countries in sub-Saharan Africa have developed very different approaches, developing new genome editing guidelines, or defining their policies in using existing laws as the basis. And before we go into those examples, it's important to note that they are taking guidance, first of all, from the Cartagena Protocol on Biosafety. And this is also something that Piet van der Meer mentioned. The definition of what a LMO is under the Cartagena Protocol is important to note here, in that it is a living organism that possesses a novel combination of genetic material obtained through the use of modern biotechnology. So an important question is really, are we dealing with an organism that possesses a novel combination of genetic material before we actually decide on whether something is regulated as an LMO or a GMO or not? So this is the definition under the Cartagena Protocol on what an LMO is. It has a clear definition of what modern biotechnology means as well. So these definitions can actually be used to come up with a country's and organization's own regulatory approach to genome editing. But it must be said that under the Cartagena Protocol on Biosafety, the guidance, there's no specific guidance yet on genome editing that is has been agreed upon by the parties to the Cartagena Protocol that other parties can use to define and shape their own domestic legislation, such as guidelines. So many countries that we have been working with are actually looking at other emerging economies, other countries that have been in this, well, have been applying genome editing for more than a decade now, and also have a regulatory approach that is has been defined and tested for well over a decade by now. And many countries are using the approach that can be found in a country such as Argentina, that after a review of the technologies at hand and a review of its legal framework dealing with biotechnology and GMOs, decided to come up with a consultative process that is pictured on this slide, where the key question is really, are we talking about a product, an application that leads to a new combination of genetic material? And this can be determined in a relatively brief consultative process to determine whether a new product should be regulated as a GMO or not, whether it falls under GMO regulation or not. And again, it also allows a country to remain consistent with the Cartagena Protocol, where this novel combination of genetic material is an important trigger for biosafety review or not. So interestingly, in Argentina, this practical approach has led to a wide range of applications and products coming out of the local public research sector as well as the local private sector, such as herbicide-tolerant canola, improved tilapia, popular fish species, as well as improved microbials that can be used as biological fertilizers. So this is just to illustrate the point that being an early adopter of both the technology as well as a practical regulatory approach leads to innovation at the country level. And this has really stimulated many countries in Africa now to define their own guidelines in this area. Examples being Ghana, Nigeria, Kenya, Ethiopia, Malawi, and Zambia, who have published their guidelines on genome editing so far, taking the existing legislation, biosafety legislation, as a starting point, as well as it's important to note that the processes to define these guidelines were also under the leadership of their national biosafety authorities. So it's really important to have this foundation in biotechnology and biosafety regulation to allow a country to move forward with a policy approach, a guideline on genome editing. And each of these countries have adopted a consultative approach to determine within a relatively short period of time whether a product is a GMO or exempt from GMO regulation. And again, with the regulatory trigger being more or less in somewhat different formulations, but the regulatory trigger is basically, are we talking about a product that has a novel combination of genetic material? Or in the case of Kenya, for example, is there integration of foreign genetic material, as they call it? But I think in practice, how they are practically applied, we're basically following the same approach as Argentina does. So this is important experience that is being developed on the African continent and where other countries, such as Egypt, perhaps can benefit from. But of course, the key is to implement these policies in practice at the national level. And there's a good example in Kenya now where a simple or a straightforward application form is available online with the National Biosafety Authority's website, where an applicant can respond to a number of questions, which will allow the NBA to determine whether a product or a project, an application, has to go through biosafety review or can be determined as exempted from biosafety review. And again, this straightforward and science-based approach in Kenya, also due to the presence of many international institutes such as IITA and others, have allowed a broad range of projects being launched that apply genome editing for agricultural productivity. Working with strong local partners, there are now several projects on the way looking at livestock applications, such as development of a swine fever vaccine or developing trypanosomiasis resistant goats, but also plant-based applications in sorghum, grass pea, sorghum, yam, cassava, and banana are on the way. With I think reviews more recently completed for cassava as well as banana. But the picture is changing so rapidly that this may need some updating. But I think the general point is clear that having this basis in biotechnology and biosafety review allows a country to move forward in genome editing as well. And this is important because in my view, the next wave of applications is just around the corner. This is an example of a startup company in Belgium, actually, not far from where Piet van der Meer is teaching, where this company is integrating advanced genomics and genome editing with artificial intelligence, allowing for multiplex genome editing, so targeting a range of traits in one new product. And this company has recently been awarded a substantial grant from the Gates Foundation to develop resilient crops with improved agronomic traits for the developing world. And this is really going to be important as we deal with heat and drought stress, particularly due to climate change. So again, this is no longer hype, but reality. If we look at some of the policy implications, those are also relatively straightforward, I think, towards the end of my presentation. We're dealing with a very rapidly evolving global context, not just in terms of regulation, but especially in terms of research and development applications, as well as actual product development, with a huge diversity in terms of the innovators, public and private sector, the traits that are being targeted, plants, animals, microorganisms that are also being subject to genome editing for agricultural improvements. We cannot find too much in terms of policy guidance from a global agreement or a global unifying approach to the regulation of genome editing. So countries are developing their own policies and regulatory approaches, using examples from other developing countries and emerging economies to set their own national goals and priorities. In general, this leads to a case-by-case consultation process that has been adopted to determine whether a particular product falls inside or outside of GMO oversight. Of course, with some important exceptions, such as South Africa. But many countries in Africa have developed their own questions and criteria to determine this important question. So this determination is made within fixed timelines, 30 days or 60 days at most. And those approaches, not only in Africa, but also in Asia and South America, are aligning with other emerging economies, especially those in South America, such as Argentina and Brazil, rather than the European Union, where a more restrictive approach has been adopted. What is important here is that we encourage South-South collaboration between Africa as well as South America, and work towards some form of harmonization in the sub-regions, as has been successfully done in Latin American countries that, with some exceptions, follow pretty much similar approaches to genome editing. And at the national level, coordination among ministries, research agencies, as well as regulatory agencies is critical, as well as local capacity development in genome editing. And that's why we're here today in Egypt. Now, as I conclude, this book was posted online last week. It's a rich resource titled A Roadmap for Plant Genome Editing. It also has chapters, pretty decent chapters on regulation as well as IP management in plant genome editing. And it's free of charge, so you can download it from the link on this final slide for further reading. And with that, I would like to conclude and thank you very much for your attention. I'll stay around for questions and comments. Thank you.
Thanks, John, for the insightful presentation. We have decided to postpone the questions until the end of the session, if you don't mind. So now our last speaker for this session is Benno van der Laan. Is he?
I was presenting on behalf of both Benno and myself.
Okay, so I didn't hear this part. Okay, so this means that we are going to move into the question session part. Any questions from the audience? Yes, please.
Discussion for all the presentation. I'm Professor Dr. Mu'tazza Omran, Dean of the Biotechnology Research Institute, and I'm very happy to share in this important workshop. And I have many questions, but I will try to ask for two important questions for me. First question, I'm going to ask Dr. Nagla, and I'm happy for her invitation for me to attend this workshop. First question, that we are going to talk about the CRISPR challenges, which is off-target mutations. And I'm, as a scientist, I'm professor of medical biotechnology, facing the problem of the mutations that the Cas enzyme is mismatching with the similar sequencing that we are going to talk about, that I'm going to edit the DNA sequencing. If it happens, we have similar sequencing. So I'm going to make editing to another sequence, similar sequence for this, so it's not working well. How to minimize and detect these mutations? And I mean here to balance between specificity and efficiency.
So this we're going to be covering the training because we already have a program and method to avoid off-target effect. This can be used by bioinformatics, by sequencing. So there is different tools for avoiding off-target mutation or editing in part that's not the target site. So all of this, the scientists already cover it before releasing anything. They have to do like the full sequence if they're going to do a mutation using transgenic or using editing. So it can be covered. This can be avoided because there is different tools to avoid, either in the bioinformatics or the wet lab in the laboratory lab. Yeah, Lina would like to answer as well.
Yes.
Yeah. So, you are very right. Like the guide RNAs which you design, the guide RNAs are the ones which are specific for the sequence of the genome where it will be targeted. But then there are some like 3 base pair sequences before the guide RNA called PAM. Those are very critical. So this is not possible that your PAM and the 20 base pair sequences will also be, these are very specific to that gene. And then when you're designing the guide RNA, there are tools. The tools can predict and say like this is your potential off-target efficiency, and this is your editing efficiency. So you have to keep the balance on that one. And the guide RNA needs to be picked, which has the maximum editing efficiency with lowest off-target. But that's not enough. That will give you based on the AI tool, right? But so in our lab, we have done this practically. So you can, once you have design the guide, so you take the PAM and the guide sequences, you blast against your sequences and you do that. But then in the end, when the product is ready, you have to also test for the target mutation, but also test for the off-target mutations. But
So you have to make tests for both.
You have to test for both before you release that thing.
Because
you don't want the off-target.
Zinc finger nucleases. It has 3 base pairs, but it's more specific but it's not working. It's too expensive, So we are using the CRISPR, which is less cheaper, the cheapest one. So it's now a present solution. It's not a future solution. CRISPR now. So it's many problems, and many scientists have this problem in mismatching.
But CRISPR is very specific. I can tell you, we have done this on banana. We didn't find any off target because when we have, even though when potentially we see this can be, but when we took them for the sequencing, we didn't see any off target. It's very, very specific.
It's problem when it happens when you have similar sequences.
So you have to be very critical in the guide RNA design with the PAM. And now there are other CRISPR tools which has actually 4 base pair PAM. So you can also go, we call them extended PAM. So you can go with that to make it more specific.
Thank you. Thank you, Dr. Nagla. Can I ask another question? I don't know. Dr. Care, the global discussion on the geriatric status of gene editor organisms. Okay. And thank you for your presentation and gave a lot of information for me. But we have here, where should we draw the scientific line between the new breeding techniques and the next generation technologies if we both both use similar gene editing tools, but they are different in, as you see from regulation. How should we draw the scientific line between both of them?
You're asking Pete?
Yeah.
Pete. Okay, Pete, can you hear us?
I can hear it partially. I can hear it partially. I could not hear everything. Could you repeat it slowly?
The question?
Slowly, yes.
Do I
please.
have to
Cannot hear
repeat the
you.
question?
Could you repeat it with loudly?
Slowly?
Could you
Slowly.
repeat the question
Okay.
loudly and slowly?
Where should we draw the scientific line between the NBT, the new breeding techniques, and the NGT, the next generation technologies, as they both use the similar gene editing tools, and how, and you know, they are different in outcome and regulations. What the scientific line that should we draw? Could you help us? Between
Myself,
both.
I don't understand your question, but I
The-
think
the
you,
difference-
yeah, yeah,
the
you're trying
different
to
scientific line
differentiate
both,
between new
both-
breeding technique, new breeding technique. You mean
both
gene
use gene editing tools.
okay?
The next generation technologies and the new breeding tools.
So did you make a difference between both of them, Pete?
New breeding technique
New breeding technique and
is using not the traditional techniques and to make the crops more faster, improve the crops. I'm talking to you, okay? And the next generation technologies, they use gene editing tools as CRISPR, as the talent, as we said in this presentation. So what the both are using the same. I'm get from information from here. Both gene editing tools. What's the scientific line between both of them?
Nagla, would
How
you like
can we
me?
draw the scientific line between them?
Nagla, would you respond to this question?
did you make a difference between next generation tools
me?
and new breeding techniques in your presentation? If there is a difference, as long as they both use the CRISPR or the gene editing. So you mentioned that they are both have benefits. So could you
Both of
differentiate
them
if there is a
using the
difference
gene
between them?
editing tools. Both of them using the gene editing tools.
Yeah, I mentioned
Happy.
that.
Okay.
So she said that both of them are using gene editing tools. What is the difference?
How can we draw the scientific line between both of them?
Why
Between NBT and NGT? And Samai.
Nagla, would you like me to respond to this?
Okay.
Okay. First, a general observation: entities are a thing. There is no scientific difference between them. But there is something important in the question here, and that is that if gene editing can have different results, how do you draw the line? How do you distinguish between them? And that is an important question. And I draw your attention to one of the slides of John Coleman, in which he had the definition of the Cartagena Protocol on biosafety, because the key question is, have I made a novel genetic combination? That is the key question for the differentiation. And the novel genetic combination is actually defined in the protocol, because it uses the phrase, and for the next slide I'll underline it, is that those are combinations that go beyond the natural barriers, physiological and recombination barriers. To give you an example, if you use gene editing to just delete one base pair, I have used technique, but deletion of one base pair happens in nature. The natural. However, if I use gene editing CRISPR template to introduce an entirely new gene, I have made a novel combination that goes beyond the natural barrier of recombination. So that is the technical line in between. So the Cartagena Protocol actually gives us more guidance than people usually assume, in that one very important phrase is that the novel combination that has been made, does that novel combination go beyond the natural physiological barriers of recombination? And that is a very practical tool, and there'll be a question of further developing and guidance as to what does it mean in practice. Is that one base pair? Is that two base pairs? Is it 20, and so forth? So I hope I've given some clarification here as to how to distinguish.
Yes, thank you.
Let me, Nagla, do I still have the floor?
Go ahead.
Nagla,
Sorry.
can you still hear me?
Yes, go ahead.
Let me add one more thing. First of all, I have to apologize. I'm traveling this afternoon internationally, so I'm going to finish packing suitcase soon while you're having lunch, and then I'm gone. But let me just give you one more thought for your discussions this afternoon, and it relates a little bit to the previous question. So if you apply CRISPR-Cas, then the CRISPR cassette is placed, enters into one part of the genome of the chromosome, and then an edit is made on another chromosome. Until up to that point, that organism is a GMO. But if you segregate out the CRISPR-Cas cassette, the elements, and you're only left with a mutation, and that is an SDN1 mutation, then it's no longer covered. Now, all of this works if you can segregate traits out, and this only works with crossable crops that reproduce sexually. If you have a vegetatively reproduced crop, then you don't have this option because you cannot segregate out the Cas9 cassette, for example. So what are scientists doing? They use all kinds of techniques, such as Krelok, to cut out the cassette that you have used to introduce the gene editing tool, but it is still there. And even if you cut it out, very often there are left tDNA borders, or there is the excision footprint. So there are all kinds of things left in those cases where you cannot segregate out. And then the question would be, what is the regulatory status of those things? What is the regulatory status of just a left border or a right border? Borders can be somewhere anywhere between 3 and 25 base pairs. What is the regulatory status of an excision footprint, and so forth? So this is, if you have further discussion this afternoon, I wanted to throw this in just to help get a bit of a pushing and pulling of the ideas, because pushing this kind of cases helps you understand your own relations. And with it, I hand back to you, Nagla, and my compliments for an excellent workshop. I'm enjoying this from a distance.
So as far as I understand, you're asking about if the plants cannot be propagated by seeds, how can we get rid of the introduced gene? I think we just covered that in the presentation, and Lina already doing this work in her banana because she doesn't use transformation with the regular method. The DNA is not introduced in the genome. Maybe she can explain it much better than me. You go ahead.
Banana is vegetatively propagated, and we know that we cannot segregate out. My understanding after discussion with some of the regulators are, if you leave the footprint of the left border, right border, they are still GM because they have 25 base pair left border, right border left. So in bananas, we don't use the stable transformation at all, so that no integration at any point. So we are using, I cannot disclose the technology, but we are using transient way of expressing the editing machinery, which is only available for short period, and that period is enough to create the mutation, but then nothing is integrated. And that's the technology. Recently, we filled the early consultation form in Kenya, and just last week we got approval, and our bananas are classified as non-GM for the field trials in Kenya. So John, I also wanted to comment on your comment. Actually, the early consultation form, individual cannot submit. So early consultation form still needs to go with the approval from IBC, and IBC submits to the NDA, and that got approved. But it's very short. It's like maximum 30 days they have to give the answer. And so you can add bananas on the list. Is approved, John.
Thanks.
Yeah,
Good news.
Yeah.
That's actually the good news for vegetatively, because in Africa that's the first vegetatively homegrown because everything was done in Kenya, for that crop.
So any other comment or question? So I think by that we can have Professor Hassan, she can handle that. Okay, thank you for all. Now we will go for 1 hour for lunch, just to give time for the people online. And okay, so I'm invited for lunch. We'll go from 1:30 to 30, then to continue with the process. So, as you know, we're going to have a discussion at the end. We were pleased to invite the people online, of course, Sheila, John, Pete. They can join us in the discussion. We can get benefit from their experience, how we can go through, and what's next in our future. This will be at the end. So yeah, it's going to be at 3 p.m. So please join us again. Don't leave. If we're just going to break for 1 hour for lunch and come back. Thank you for your contribution, everybody. Thank you.
Thank you. Bye-bye.
Hi. Ladies and gentlemen, the lunch will be using the elevators at M level if you want to reach to the main restaurant of the hotel. Enjoy. Bon appetit. لديه شاحن. الرجاء التوجه إلى قسم الريجستريشن.
Dr. Sheila, are you online? Dr. Sheila.
Hello, good afternoon. Can you hear me?
Yes, we can. Sorry
Great.
for the delay.
No,
No, how people behave after lunch.
that's fine.
So let's start.
Okay, let me see if I can share my screen. Share this screen. Am I sharing the right screen? No, I want to share this screen.
That's me.
I always find it a little bit difficult whether to share screen or window.
Yeah, we can see you, but we don't see the screen.
Can you see?
We can see you, but we don't see your presentation.
Oh dear.
Sheila, try to share your window, not the screen.
Sorry?
Window, try to share your window and select your file.
Okay, this one.
Don't
Oh
share
yeah,
screen.
this one. Yes?
It's
Yeah,
very
that's-
hazy.
Quality is
Yeah,
not
it's
good.
coming.
I think your connection
That's it?
not good.
The resolution is very low.
Is it?
It is.
Oh dear.
Can you change your connection, Sheila, if you have another connection? I think you have the connection is not good.
No, I don't have another connection.
Okay, if you can turn off your camera to save some bandwidth.
Okay. This one. Am I sharing? Oh dear. Would it help if I sent you the presentation?
Yeah, okay. If you can send the presentation, it's fine.
Let me just do that right now. Slightly different than the one I sent earlier. Nagla.
Sheila, you will send the presentation.
Yes, it's uploading. It's a large file because it has lots of pictures. So I'm sending it right now to Professor Nagla.
Okay.
I have sent it. Send. Right now. It has gone. There you go. Okay.
Dr. Sheila.
Hello.
That's Sheila. Did you send it?
Yes, it's sent. I've
Okay.
sent. Yeah.
We are waiting on Professor Chandler.
Okay.
Email.
Thank you.
Yes, okay.
Dr. Sheena?
Okay.
Yeah, we're almost there. Just loading the
All right.
presentation on the laptop. Yes.
Thank you. Great.
So can you see the presentation?
Yes, I can. Wonderful. Thank you so much.
Everyone move between slides, just let us know.
Yes, that's fine. Okay.
Okay, let's get started.
Great.
So our next and final speaker for the day is Dr. Sheila Obem. She is the executive director of the Alliance for Science Global Consortium. Dr. Obam is a distinguished scientist and communicator who bridges the gap between complex laboratory innovation and community level adoption. From her early work on genetically modified viruses for pest control at Oxford to her current occupation, Dr. Obam has dedicated her career to the responsible use of biotechnology. As a global leader in science communication, she heads international efforts to counter misinformation and ensure that innovations like gene editing are met with public trust and science-based policy, particularly across the African continent. She is an avid believer in inclusive innovation, insisting that local knowledge and community trust must be placed at the very center of how we introduce new technologies to farmers. Dr. Obam, please go ahead.
Thank you so much. And it's such a pleasure to be able to join you today. I thank you so much, Professor Nagla, who has been a real colleague, friend, sister, collaborator for many years to Alliance for Science. And we're very happy to have an opportunity to join with other partners and colleagues in this very, very important workshop. Yes, communication is very important, and I'm the last speaker after lunch, so I hope I'll be able to create an engaging space to precede the discussion, which I think ends this particular day's sessions. So if you'd like to go to the next slide. So in the next slide, I just say the same thing. The background is that communication is essential. It's not an add-on. I know because I trained as a scientist, a laboratory-based scientist, and it took me a long time to value communication. But I found that we couldn't do the work that we needed to do in the labs because we hadn't communicated the science properly. And this was when I was in the lab over 25 years ago, and the EU banned GMO research. And my career took a shift because we hadn't communicated or engaged with communities well. And so communication means not just sharing, but it also means showing, teaching, transferring knowledge about discoveries and observation, and not just to colleagues within our own echo chamber, but to society at large. And so good communication must accompany science. And I'm sure that you find that in your work, all of you as well. Next slide, please. Let me introduce the Alliance for Science for those of you that don't know us. We actually were founded in Cornell University in 2014 by Professor Sarah Evanja, who was working then with the great Professor Ronnie Kaufman on the wheat rust project that was funded by Gates. So it was a big 70 million project on wheat rust, and they were meeting a lot of resistance from anti-GMO communicators on campus and around the world. And so Sarah had the wonderful idea that why are all the science voices so silent? Why don't we empower scientists and those that are pro the science to speak up? Perhaps they don't have the tools, perhaps they just need some push. And so she got some funding to start Alliance for Science in 2014. And that has been our mission since, to promote science-based communications, solutions through advocacy, communications, and research. I took over in 2022. And we focus our training on scientists and building capacity on ag biotech for champions in Africa, Asia, and Latin America. Alliance for Science was basically began with training those champions. And so they would come to Cornell for 3 months, be trained on science communication and given some support to go back to their communities to do some work. And there were a wide range, not just scientists, we have priests, we have students from over 40 countries, all age ranges, young, old, farmers. It was very successful, and we also added executive leadership programs that were shorter than the 3 months residential in Cornell. And at that time, we focused our initial advocacy on advocating for GMO crops in Africa. And since then, we have over 1,000 specialist tools and resources in many languages for different stakeholders. And we collaborate closely with the CGIAR system, especially those on the ILRI campus, IITA, ICRAF, many academic institutions, national agricultural research institutes globally, and global North partners, Cornell, Michigan State University, UC Davis, University of Ghent, have been very supportive, and Tuskegee University, just to name a few. But we've practically worked with everybody in the ecosystem around ag biotech globally. Next slide, please. This is just a synopsis of what we do. At the heart of it, empowering, mobilizing community advocates. Our biggest team is in Africa, doing the communications and research and implementation work. Our research focuses on looking at uptake, sentiment analysis. How are people viewing our messaging? Who's listening? What are they saying? And our training tries to adapt as quickly as possible to that. But we also work with regulators because they are very important in building trust. So working with the national biosafety agencies in each country is key to it. And we had a small team led by Pablo Orozco that he was also a negotiator on the CBD COP. And so we were also working with those negotiating at the front lines of policy decisions. So that's the key. Communications around GMO, gene editing more now, and in South Asia and Sub-Saharan Africa. Thank you. Next slide, please. So our core crops or products that we got our grants were mostly funded by Gates Foundation with other donors and supporters around the world. But we mostly spoke about BT cowpea, Wema, BT brinjal, have a sub-grantee, Farming Futures Bangladesh, that was really working to build support around golden rice. Banana, we have worked with Professor Lena Tripathi, who is a skilled communicator and is one of the best advocates around many of these crops. So we work also around cassava and potato. Alliance for Science in Rwanda, led by one of our fellows, have a grant right now to work with SIP around potatoes. The technologies we look at, GMO, gene editing, precision breeding, gene drives also. Target countries, you can see there, mostly Africa, but also Bangladesh, India, Philippines, Indonesia. But we also keep an eye out for influencer countries, USA, EU, UK. Next slide, please. So that's a picture of our team at CBD COP15, and they're looking at creating like-minded groupings in member countries across Asia and Africa who are really looking at the text. So coordinating how we can bring more advocates at the front lines of policymaking. Next slide. A lot has been said about the regulatory landscape. This slide is what we would normally use in our communications. So it's probably a little bit out of date, but it's just to show the gaps in discussions around gene editing and what the continent of Africa looks like, and why really we need to
Sheila, are you there? We can't hear you, Sheila.
oops. Okay.
Okay,
Next
we can hear you now.
Okay. Sorry, next slide. Thank you. We recognize that we have to have an understanding of the nuances in regulatory approaches so that our communication does not bring in errors. So we've largely lumped, put them into 4 categories when we're looking at gene editing. Category 1, where GMO regulations are applied as is to gene edited products. So that's one category of regulatory approaches in certain countries. 2, simplified GMO regulations, which is applied simplified GMO regulations applied to gene edited products. 3, where they're exempt, gene edited products are exempt from GMO regulations. However, confirmation by government is required before placing on the market. Then category 4, confirmation is not required. They're exempt from GMO regulations, and but prior confirmation is not required. So those are different approaches that inform our communication approaches. Next slide. So yes, when you look at those 4 approaches, when we are looking at training and looking at our messaging, where's the middle ground? We're always trying to find middle ground messaging because all messaging is global. In the end, the context matters, but you also have to find a generic messaging that will also allow all those approaches to be understood and to drive understanding in those countries. Next slide, please. So what can we do? Next slide. Our job is, our mission is to create an enabling environment. We are here because we're like the solution in the Petri dish that allows the organism to thrive. That's what communication is. We want to support to create an enabling environment for researchers, policymakers, and the community, public sector, civil society, farmers, so that science is prioritized through policy decisions to make sure innovations are fostered and supported, and looking at societal, legal, and policy spheres to, in the end, facilitate adoption so that products come to market, but then also that people appreciate them and use them properly. Next slide, please. So in 2022, we had a grant that was funded by the Foundation for Agriculture and Research, FAR, to determine how the US feels about gene editing. So FAR was really looking at a difficult regulatory environment then in 2022 and wanted to assess how messaging could help, how we could develop a toolkit that can then train scientists and support the policy work. And so I'm going to present some of that research. So it's very US based, but it informed how we developed our toolkit, which it's transferable. So focus groups were set up across the different regions in America, and online surveys and a lot of desk-based research. Next slide, please. So the question then, and perhaps now, is why Gino? Why does it matter? Why should you bring more innovations on board? And so we clarify that it's a method of selective breeding which makes precise, intentional, beneficial changes to genetic material of plants and animals. And the stakeholders we are championing because we believe in its efficiency and its abilities to fast track productivity. We also think it gives lots of opportunities for innovations to come on board that are less controversial. And we work hard to support the EU to see this new way of doing things. And I'll talk more about that later. Next slide, please. So the science communication problem is about how do you get people to look at the facts and pay attention to them in making decisions. some scientific phenomena like photosynthesis, there's no controversy. People accept it. It is what it is. There's no much discussion about that. But then when you come to things like climate change, vaccination, GMOs, there's heated debates. And that's because at the end of the day, unless the scientific findings align to an extent with people's personal beliefs and their value systems, there will be polarization. And so that is really largely why this idea of the deficit model, people don't accept because they don't know, doesn't work. People can know and still not accept because scientific evidence does not quiet the dispute because there's a misalignment with values and beliefs. Next slide, please. So it's the whole heart and head issue. Your head knows, but your heart doesn't accept. So how do you get congruence between the head and the heart? You need to encourage open dialogue, and that's what our values-based messaging toolkit ultimately achieves. Go ahead. So we start off by listening. We listen at many levels. We listen by searching the data. We invested in support from Cision Prime Media, who are one of the largest global media monitoring firms. And they created a dashboard for us that looked at measuring market trends and sentiment on gene editing. And we use those to develop specialist media monitoring and community engagement tools to continue gauging sentiment and support on gene editing. And we adapted those tools accordingly as we went along and began to engage communities in their own languages, developing our materials with a view to local context in Kenya and Ghana, Nigeria, and looking at cultural beliefs around food and food systems. Next slide. So here's just a sample of what, just one slide from a large report I would get every quarter. We'd look at all media globally, and we'd also look at local media, traditional social media, and we'd gauge sentiment around different topics. Next slide. And it would help us to say, okay, state of the media is, and here's an example, gene editing at that point, this was an important moment when gene editing conversations surpassed conversations about GMOs, and people also began to talk about gene drives, and we began to see raised favorability around gene edited crops in different high reaching media outlets. And we began to see that the public were beginning to be more interested in these biotechnologies and in these emerging areas. And but the risk of that was the conversations around GMOs were reducing, and much as we value the rise in gene editing conversation, GMO conversations are also still important because it's still a valuable technology in our toolkit. So we also pay attention to that. Our people understanding GMOs and gene editing are part of a suite of solutions that are open to scientists to tackle a range of problems. Next slide, please. So we would get a slide like this that would tell us which media are reporting on these keywords. So you got there in that particular slide, you'd see Chinese media, CNN would be reporting highly, BBC further down. And we'd also get keywords around the crops. Which kind of crops? They talk about cotton, are they talking about maize? So we look deeply on where the conversation is happening, who's having it, and then how do we engage? Next slide. So out of all that, we got some key points for communicating gene editing. We recognize now that consumers were interested in learning about the role of gene editing in making food more nutritious, improving animal welfare, reducing pesticide use, reducing water use. And those that were negative were still, as you would expect, worried about safety concerns. They wanted more information. They were concerned that it's not natural. This is a big trigger word. And also that it could be misused, that this technology can be misused and empower a few members of the community, or big ag. Next slide, please. But those that recognize beneficial uses, and I'll come back to this again with more examples, recognize that it improves yields and resilience of crops in regions that have greatest challenges of hunger and climate challenges. It reduces pesticides. I guess a new point here is that it could reduce the price of foods because it increases supplies, and affordability of food could rise, as well as helping the environment. Next slide. So those that were interested, respondents also identified applications as desirable uses of gene editing in agriculture was around biofuels made from plants and algae and bacteria, and realized that this could reduce our need to burn fossil fuels and import fuels from other countries. So this balance of new innovations and technologies empowering local communities was very positive sentiment. And also that microbes that could degrade existing plastics in our environment, that also came up, and that it also reduces the time needed for plant breeding, this acceleration of even innovations. Next slide. So this is very America-based, but it gives you an insight. Those who supported it were more likely to already be people that read a bit about science and technology every day from traditional and scientific journals. They were more likely to be educated with higher incomes. They more likely lived in a city with children in their household, and they were more likely to be men and Democratic voters. This is important information in America. Next slide. And those that were resistant were those that were unlikely to consume any media related to science and technology. They tended to be less educated, lower incomes, lived in rural areas, tended to be women, and Republican voters. Next slide. Those that were neutral dipped in and out of consuming science and technology media. They were less educated, likely had no children, lower incomes, lived in the Northwest of America, were likely to be women, and also ambivalent about voting. So they were not committed to one party or another. Next slide. So it was important for us to realize, to do this research, to recognize our audience, even in the American context. Those that were positive are potential allies and people that we should try to engage with and produce content for. Those that were neutral, we should also engage with and add supplementary materials for them to understand more. But those that were resistant were likely to have already made up their minds. So anything that we did was unlikely to shift their position. So it's also about how do we balance our resources and who do we engage when and how. So that was, we had an audience map in, and you always have to do that in all science communication. You must begin with who is your audience and how are they segmented. Next slide. So our approaches to communicating gene editing. Next slide. So the first approach, we focused on public benefits and values. Consumers were curious and generally supportive of the science. and wanted to know how it could benefit the environment, make food more nutritious, resist disease. But an important point to make here is in communicating with them, it was important not just to focus on it improves yields. Improving yields is great, but generally consumers want to know that you're producing enough food, enough good, healthy food with less input. So producing enough with less. Yes, yield increase is fine, but they're seeing that really is something that industry cares about. But overall, how is the balance of production and inputs and impact? Next one. So messages about gene editing should focus on practical applications, both in agriculture and the real world. So always a practical anecdote. Next one. Yeah, people want to know about gene editing is making a positive difference in achieving food security. And I think I've said all this before, but I'll just reiterate again, lowering food costs. Next slide. So an example, as you heard earlier this morning with Professor Lena, talked about disease-resistant banana, really important in Africa, food and cash crop, and people can see it walking around their community, the diseases that kill the crops and the bananas, and they can understand how it supports the security, food security and farmers' livelihoods. Next slide. Next one. Drought-tolerant wheat. People understand the economic value of wheat. And they also understand that in producing this, we can reduce water use, and so it can survive in dry environments. So they're positive about its application in developing drought-tolerant wheat. Next one. And chocolate. So we talk about healthier chocolates. Scientists in Colombia using gene editing to alter the root system of the cacao plant so it doesn't absorb heavy metals from the soil. So healthier chocolates resonates well with the public. Next one. And you heard this example, I think, from Dr. Komen. He talked about in Japan, 2 types of fish that were proved gene edited, they grow faster, so reduce food cost, and they reach market weight faster. So on the same amount of food and lowering production costs. So that's a good example of producing enough with less. Next one. So the second approach is you connect it to human health and environment. So how does it produce healthier populations and reduce disease resistance? So yeah, human health and environmental challenges. I think I've said this before. Next one. Yep, next slide. I think I've said this all before. Next slide. So yeah, reducing pesticide use, fertilizer use, and so language around that is scientists are using gene editing to help cereal crops like wheat, rice, millet fix their own nitrogen from the soil like beans do. This could eventually replace their need for nitrogen from chemical fertilizers. And controlling plastic pollution. Researchers are using gene editing to engineer an enzyme that accelerates degradation of plastics, helping to address the persistent global problem of plastic pollution. Then greener sugarcane. Scientists are using gene editing to fine-tune sugarcane. This could help reduce the environmental impact of growing sugarcane, which is used to make bioethanol for biofuels and plastic that is 100% recyclable. So this is some of the messaging. Next slide. So the key points are share the benefits most likely to consumers, not just food producers. Share the benefits to the environment. Share the benefits to animals, how it improves animal well-being and reduces disease. Share the benefits to plants, like tolerance to disease and weather conditions, not just increase yields. Next slide. and also have real scientists in the field showing a passion for their work and showing how their work actually directly solves a problem. Like here we have a quote from a Nigerian crop scientist, Sylvester Okio, where he talks about, if no food, you have hunger. GM maize can increase yields and prevent hunger. And coming from him directly has a lot of impact because people see their scientists doing the work to solve challenges that they can understand. Next slide. When consumers were asked who they trust most for information about food, several studies revealed similar findings. Healthcare settings are always good places to have a conversation about difficult technologies to gain trust. So doctors, clinics, hospitals, that's actually a primary source of where people have found out information about gene-edited foods in the US. And respected organizations such as FAO, USDA, FDA, WHO have high trust, as well as regulatory authorities. And we found that when we engage regulatory authorities in any of our communications or showed that they were part of the process and how, it increased trust. And when we didn't, it reduced trust in how people viewed the message. People want to know that somebody has oversight. And so it's important to get different voices with different credential, experts, farmers, and actual consumers. Next one. So as I've said before, the key questions, and you've all heard, is it safe? Is it okay to eat? Who did the testing? Is it being regulated? How long is it being used? What are the downsides? Now, this is important. We have to also show people that it's science and science has risks and it's not always a positive story. Otherwise, you don't build trust by just telling one side of the story. And lastly, people also want to know, how can they tell if a product has been gene edited? And because they're not always labeled. And that's a question that you will come across that you would have to answer. Next slide. How do people want to learn about gene editing? People like video. They like stories about farmers and scientists. They want to see real people doing real things. They want human interest stories. They want to hear it on radio, especially rural communities. Radio is still king across Africa. They want free samples of food. And in America, we had a food truck called the Modified Food Cart, and we would take it along and have slices of food, and people would taste it. And that's being done by colleagues across Africa. The eating is believing, is also seeing is believing, as well as virtual reality tours of labs and farms. And next slide. What is not preferred? Apps, social media, infographics, videos. Social media now has decreasing trust, and people don't want to learn about complex technology. They perhaps will check it to affirm it, but that's no longer a key reference point. Next slide. GMOs and gene editing, yes. People still have reservations about GMOs. Nearly 2/3 question the safety of eating GMO foods. And it's important that you understand the tension between these two feelings. And we found that when we explained to communities or people the difference between GMO and gene editing, it actually decreased trust because GMO is such an emotive trigger for those that are anti-GMO. And so when we're talking about gene editing, we tend to just specifically focus our messaging on gene editing and what it is, and have a separate conversation. But putting them side by side actually sometimes does not help uptake of our messaging in a positive sense. Next slide. I won't go into that, but that's just to show you that we have to train people, especially lay people, on the difference between GMO and gene editing, and they have to clearly understand the language around that when they are engaging. And so we put a slide like this and spend some time when we're engaging with non-experts and not people like yourselves. Next slide. So helpful analogs that we give them, if you press again, is yeah, next slide. Yeah, it's like if an organism was a book and the words within the book are the genome, then gene editing is like changing a couple of letters in the book. Press again, next slide. But genetic modification is like when you bring a whole new paragraph into a book that was written by a different author. So that's one analogy. Next one. Okay, press again. So another one is around talent. GMOs are like outside talent that you bring in to help you do something really well. But gene editing, press again. Gene editing is more like bringing the best out of what's already there. So those are simple analogies. Next slide. So we encourage people to use positive practical emphasize positive practical applications. And in this workshop, I've heard so many and done so well because everybody is presenting here as an expert communicator. So that's an example of how you look at the practical, emphasize positive practical applications, but also engage scientists and farmers as messengers. We have a significant group of farmers that work with scientists as key messengers. And so you can put them in the same room, in the same workshop, and use content from there to tell messages differently and create powerful storytelling techniques. So storytelling is a medium. But lastly, and very importantly, address questions. Make sure you think about the questions that you're asked and try to answer them. As I said before, everything is not positive. There are some negatives, there are some flaws, there are some limitations, and there have been some mistakes. And in terms of how people use the technology, of applied the policies. And so don't shy away when challenged, but try and engage and read up on that. Next one. So as I said before, don't mix gene editing and GMOs, and because of the baggage that come around GMOs in the same conversation. But at the same time, don't throw GMOs under the bus. What we like to say is it's like having a toolkit for your car in case your car breaks down. You may have GMOs are like that one tool that you don't use often, but they may come a day that you may use it. So it's a technology that we will have to use. It's a plant breeding tool that is still going to be useful for us, and it's part of our suite of technologies that scientists use. And we are not saying it's no longer relevant now that gene editing is there. And don't hide your use of gene editing. We say to producers, there may not be labeling needed, but that doesn't mean we can't talk about the foods that have been gene edited, because it should build trust. And as I said before, we can't rely just on social media for messaging. And lastly, don't use GE as an abbreviation, because that confuses people, because it's also it confuses people because they understand genetic engineering is also GE. Next slide. So yeah, coming to the end of a presentation, we're really looking at our values-based communication toolkit that we developed out of this research. Next slide. So as I said, values-based communication is because we've realized that it's a strategic approach to sharing scientific evidence without unnecessary polarization. You are creating messages that connects with your audience shared values, attitudes, outlooks, goals, and worldviews. And it differs depending on who we're training and where, but I'll show you two examples and you see what we mean. Next slide. So as I said, we developed a toolkit to combat misinformation, and we use it as a training tool, and we help individuals and communities create value-based messages that connect with their audiences. And the training can take 2 or 3 days, but we have done it over a day. We've been working with African Plant Breeders Academy, as you heard of the CRISPR-Cas course that has been, the trainings going on in ICRAF. So for 2 of the cohorts, we did actually also support the cohort with a short introduction on the gene editing toolkit. And so they began to think about science communication. But we had 2 or 3 day training with students in America, Tuskegee, and we had a 1 week, well, not 1 week, 5 days training. Well, there was a 5-day training in University of Calabar, and we added 2 days of science communication training on using this gene editing toolkit in 2024. Next slide. So here, so you're looking at your audience. What are their values? What is the core message that you want to pass through? And how do you integrate the values to your core message? And then what channel do you use to reach your audience? Next slide. So here's an example. Your core message is, our regulatory system is based on sound scientific research. And you're talking to a group of mothers. Their value systems, the audience values family, safety, health, and the intuition they have about what's good and what's not good for their children. So how you would turn a message like our regulatory system is based on scientific research to a value-based message, you would say something like, moms have a sixth sense when it comes to protecting their children. And that's why our moms sleep peacefully, knowing that nothing gets approved in our office without rigorous scientific evidence. And here now, you're showing your audience that you believe that they also come with the knowledge, their intuition, and that you understand the emphasis on family and safety. So that's how you turn a generic message to a very specific message to a specific audience. Next one. So here you are looking, your core message is genetic engineering is safe and beneficial technology. And your target audience values tradition, human rights, local participation, sustainability, and community. So your messaging would now be nuanced and expanded to say, this technology coupled with traditional knowledge can help us strengthen our native corn varieties, land races, and their potential for adaptation to climate change, while contributing to reducing our dependence on corn imports. Our indigenous people have provided guiding principles for research into improving local varieties. They work tirelessly to harness the power of nature to ensure prosperity for their communities. Our scientists are proud to follow in their footsteps. So here you're showing respect for tradition, empowering self-reliance, and that you're part of a community and you're looking at sustainability. So these are examples that people work. So you can, when you do this training, you are then asked to come up with about 5 to 10 value-based message based on the audience that you face. And you would then go through this process to then develop your value-based messaging. And that value should not be forced. So if you're not a person that really values traditional knowledge or indigenous people, you shouldn't just say so because it's good messaging, because it wouldn't come across as authentic. Then we would develop a different way of triggering that recognition. So this one is an example of something we came that came out of a workshop of those that do value indigenous knowledge alongside the science and technology. Next slide. So as I said, we developed that in the US and worked closely with the US and have started to test the toolkit with African communities. We haven't done many workshops with different groups across Africa, so I can't give you many examples, but we did form a South-South collaboration, like-minded group, as said, led by Pablo Orozco, and did a workshop. And these are mostly regulators and policymakers from 14 countries across Africa and Asia. And we brought them together to begin to think about how do we work together as those at the front line of change in terms of policy making. And so I hope in the future that this group, we can, when we're able to get some funding, to begin to expand how we work to support value-based messaging, understanding, so that they can use it in their work and with their institutions and their community. Next slide. And lastly, just got a few more slides left, I just want to highlight what we have to do. So I've talked a lot about the pull factor, value-based messaging, where you're bringing people closer and you're trying to share common ground. But at the same time, let's not forget that there is misinformation and it's rising and it's like a tsunami, quite frankly. And now with AI and AI generated content, really clever influencers and activists are really challenging us in our science communication, and there's a lot more that we have to do looking at the environment that we live in now. Next slide. So an example of some of what we've done, we do research periodically to engage the media in at least understanding how they could either unwittingly or proactively be sharing misinformation. So we did some research on GMO misinformation in the Kenyan media. It always rises just before court cases. And you have the activist groups are very good with social media, very compelling. And so we had to engage the media to say, by the way, there's a lot of misinformation in your content. Are you aware? Because generally we found that they don't want to be spouting misinformation. They want to be good journalists. They want balance. They want support to present balanced viewpoints. Next slide. So we did a quantitative analysis. We actually looked at all their articles that year. We looked at where misinformation was unchallenged, where it was challenged, where it was fact-checked. And we said, listen, you need to improve your fact-checking abilities, where you also use misleading images. And largely that was, it was a very positive entry point to a conversation with the media, traditional media this was. Next slide. And also we gave them examples, because a lot of these examples were what you call clickbait, because some of these are mostly politicians quoted, said something and it was misinformation. And the press just shared it without really challenging it. So we showed them examples of where this is really damaging, like when Professor Wajakoya said that GMO foods, Kenyan men could grow breasts and women could grow beards if they ate GMO foods. So we make a point of challenging this, not directly with those that have said it, but with the media outlets who have a responsibility to be fair and reasonable. Next slide. And As I said, EU is an influencer country, so there we used a different technique. We worked with the Breakthrough Institute to look at the cost of non-adoption of genetic technologies. And so they did an analysis and came up with the number 3 trillion is the cost of saying no. This is 3 trillion is the cost to the EU of non-adoption. And we launched this report by the European Parliament before a key debate, and it got a lot of traction and interest and allowed us to make an argument that is an economic argument, not just about feelings and sentiment. Next one. And so we repeated that in Kenya recently, where we also looked at the cost of delay on uptake of genetically modified crops. So there we worked with OFAB, AATF, ISA, SIF, and other partners. And Breakthrough Institute helped us do another report around the cost of delay, and we launched it and did a lot of work around what is the cost of saying no. There is a cost of not taking up these technologies. Next slide. I think it's my last slide. Yeah, that's just the cover of the report, which is available online. Next slide. Yeah, so thank you very much. I hope we can connect in other ways. As I said, AI, this is the age of AI and the age of misinformation. Whatever work you're doing, you're going to need good science communication to create and to keep an enabling environment for that work to reach its true beneficiaries. Thank you very much.
Thank you, Dr. Sheila, for the nice presentation. So now we'll convert to the discussion. Please, I would like us to invite the speaker. Please, can you come to the table to start the discussion? The speakers, please.
The events, finally we reached our last part of the event today. So we would like to have an open discussion about what's going on and what we take over today. So we're going to raise a different topic and we open the floor for discussion. And we have people with us through the internet. They can share with us about what's going on, what next, what the future is going to be. How can we help each other to reach a compromising conclusion? How can we push the application of gene editing and so on? So first of all, maybe we'd like to talk about how can the institution collaboration integrate in the leading transparent public dialogues. So that means that we need to have an internet connection with the different institutes. So if you would like to talk about it, whoever would like to discuss these parts, I'll start maybe with the Academy of Science because they have the network in the center.
Thanks to Nagla. We are open for the cooperation in different case, we can talk about the co-funding mechanism for the funding. We can allow the co-fund mechanism between the SRP as a fund agent and between